Hello and welcome to Chapter 19 Genetic Technology 5. This is Bioinformatics. What is the function of bioinformatics? It is for the collection, processing, and analysis of biological information and data using computer software, which kind of explain the name bioinformatics, basically biological information and basically a field that studies biological information, not just the collection of it, but also the processing and analysis. And that is why this requires many different fields, not only the basic bio and chemistry that's involved, but also math, stats, as well as computer science and engineering in order to process and analyze those data using computer programs or software. Now this here is the mark scheme answer, but in a very short version would be that bioinformatics is dealing with databases. It is a storage of information, it is a database of information, and basically, actually, in real life, we call this big data. Okay, there are huge quantities of data from all over the world. Most of it is already collected. So there's a lot of DNA sequences, basically there's a lot of data from microarrays even and other techniques. And yeah, all this collection of data is posted online in a huge database. So you can actually now search online for DNA and amino acid sequences and you'll be able to find them. It's available to all, but data is not meaningful if it just remains as data, right? You have all these sequences and all these amino acid sequences, but you do need to make data meaningful in order for it to be useful. So if we can take this DNA amino sequences and be able to compare them across maybe different species or different individuals, we can then model or predict the tertiary structure of the protein and therefore may be able to understand better of what that protein does and how it functions, where is it found, and use that information in order to develop drugs, develop cure for diseases, or just understand more about our processes. So yeah, it is a huge field and it's very exciting. You just learn a little bit though, it's just a very small part of your syllabus. Now before we go on into more detail, I just want to talk a little bit about DNA sequencing. It's something that I have mentioned a lot actually for like the past few chapters, right? DNA sequencing is basically a tool to determine the exact sequences of bases in genes. The methods for DNA sequencing is no longer in the syllabus. But I just want to say that, hey, DNA sequencing used to be very difficult. It used to take very long periods of time. And the machines needed for DNA sequencing would take up an entire room. But right now we have advanced so much, and the technology keeps evolving. So right now you can even sequence DNA using this small device here that can fit on the palm of your hand, and you only need a very small amount of DNA to do that, and it is connected to your computer via USB. So it's not complicated at all. Yeah, anyways, again, you don't need to know the method, you just need to know that it is used to determine the exact sequence of bases in genes. It is now a fully automated process. A lot of the genomes of many species have been published, and this gives us information about locations of genes, and as well, it provides evidence of evolutionary links between organisms as well. So yeah, we do a lot of the DNA sequence. There are very essential information here. So essential that one of the first projects when it comes to DNA sequencing was the Human Genome Project. I think this started in the 20th century. I don't remember the exact year, but you can search it up. And basically this project, the Human Genome Project, was the first few DNA sequencing projects ever, and it basically sequenced all of our chromosomes, all 22 of them, plus X and Y chromosome. And this gives information about the location of genes. And through this Human Genome Project, we finally found that, hey, actually, not a lot of our genome, a lot of our DNA actually doesn't code for protein. So they kind of sit around, and maybe they are regulatory sequences, so sequences like promoters, like that are specific DNA sequences for other proteins or transcription factors or even enzymes to bind. And this actually helps in regulating gene expression. So yeah, Human Genome Project has been a huge breakthrough in this aspect and brought about many different research and has contributed to many of them. Okay, anyways, that is DNA sequencing. What do we do with them, with the data? We basically, all the data in DNA sequencing, along with microarrays. In microarray, there is like 10,000 cells, right? Every time you run the one microarray chip. So every microarray chip has 10,000 cells. 10,000 cells means 10,000 genes. So every time you run it one time, you get information on 10,000 genes. That is a lot of information because you're likely, as a researcher, to run it a few times. So you end up with a lot of information, aka big data, and we kind of put them into the database, upload it, and that is actually not the hard part. The hard part is prediction and analysis, and this can be used in modeling relationships, so seeing what proteins interact with each other, what genes are expressed when together with other genes. We can use this to compare between DNA sequences as well as amino acid sequences. We can use this information to predict what happened in evolution, so evolutionary events, to see the relationship between different species and even discover new molecules. There are many proteins out there that we don't know yet, but through this process we can figure them out and predict, again, the 3D sequence. Now there are a few databases that are available here, and if you do my quiz, I actually help you use UniProt and BLAST a little bit. But I've left some links here, you can go and check it out. I'll put them in the description below as well. Anyways, that is bioinformatics. If you didn't hear what I just said, basically it is a collection, it is processing and analysis of biological information and data using computer programs and software. This definition is very important. Now, what is it for? It is for, number one, as I said just now, identifying evolutionary relationships to find genetic relatedness. We use bioinformatics in order to find evidence for evolution as well. So if you don't remember what this is about, you can refer to my video two chapters ago on the evidence of evolution. But basically, what that slide says is that the closer the similarities in DNA sequences, the more recent the common ancestry they are. It means the closer the similarities in sequences, the more related those two individuals are, or those two species are. Now, what other applications do we see in bioinformatics? We can compare between genes or proteins of humans and model organisms. Now, what are model organisms? Model organisms are organisms that we use very frequently in research, okay, for drug testing, for research of diseases. And yeah, these organisms are mice, so we dissect mice a lot and use mice a lot in experiments, right? Drosophila, fruit flies, yeast, C. elegans roundworm, these are model organisms, very widely used in research. We understand a lot about them. And we understand even more about them now because of bioinformatics. Because we can know the genetic sequences, the genome of each and every model organism. And therefore, we can figure out, okay, if we're studying a target gene, we can figure out which model organism has a similar sequence and protein have similar structure. And if they are similar, then they can be used to investigate when and where genes have an effect in humans. So whatever conclusions you can draw in the model organism about that particular target gene can be then also extended, extrapolated maybe to human beings. So yeah, so bioinformatics have really helped us with that. Number three, last but not least, it also helps us identify genome sequence and proteins of pathogens. So this is very important, like COVID-19, right? Bioinformatics has been essential to track like how how COVID-19 has mutated, okay, and also contributed in vaccine and drug development. By knowing the protein sequence of a particular protein in COVID-19, we can then find out the mRNA sequence and use the mRNA sequence as a vaccine. That's crazy. Anyways, I would love to talk about that, but this is not about that. You can search it up. I think I've linked a few links to the mRNA vaccine for COVID-19 in the Q&A channel, so you can check that out. But for now, let's talk about Plasmodium. This is actually in syllabus. Let's talk about the one in syllabus, yeah? So drug development for malaria. Malaria, again, is caused by Plasmodium. You can use the database to identify the genes in Plasmodium to predict those amino acid sequences of the proteins based on the gene sequence. You can predict the 3D structure of proteins in Plasmodium and identify functions of proteins from the 3D structure through computer software, okay? And if you can do all these, basically, for Plasmodium, then you can formulate drugs to target that particular protein or enzyme which is most suitable. You can block the activity of protein enzyme, and maybe that would block the pathogen Plasmodium from doing whatever it does, okay? And maybe we can develop drugs to disrupt structure of protein enzyme or even prevent the transcription of the gene coding for that protein in the first place. So what we are saying here is you can get a lot of information just by analyzing the genetic sequence, and then you can use this knowledge you have gotten here in order to formulate drugs that targets every part of the process. So blocking the enzyme itself or preventing the transcription of the gene, maybe even blocking other proteins which enhance the function of this particular protein, using many ways to block all the processes in order to cure malaria. Now, the previous like project advisor I've worked with actually is doing this drug development for malaria thing, but nothing is on the market yet. It's still in research. Pretty cool. That's a conversation for another day. But with that, we are done with bioinformatics, honestly. Before we end our syllabus, honestly, I just want to talk about one more genetic technology. So this is extra, this is not in syllabus. So you can pause the video now and close the tab if you want to. But if you're interested, please stay on. This is about genetic editing, genome editing. Now, in the past few videos, I'm not sure which one anymore, I've said that it's incredibly difficult to place genes to insert a gene in a particular location. It's very difficult to delete or introduce a mutation in a particular specific location in a chromosome in a live organism. Okay, it's very difficult. But however, a recent genetic technology has advanced, and some researchers found this brilliant enzyme called Cas9 and developed this technology called CRISPR-Cas9. CRISPR stands for this really long thing that I'm not going to even read. Okay, it doesn't matter. And Cas9 stands for CRISPR-associated protein 9. Now, this technology allows you to cut DNA at specific loci and allow genome editing. It is inexpensive compared to other methods. It is much easier, it's very elegant, and it's very effective. It is initially found in the bacterial system used to protect itself against the virus, and this is how it works. Now, there are a few components here you need to know about. Number one is the Cas9 enzyme, which we talked about just now, and what it does, it cuts DNA. The second component you need is a gRNA, guide RNA, that is complementary to the target DNA. So, for example, you want to insert or delete a gene at this particular target sequence. What you need is a guide RNA with a complementary sequence to the target DNA, and it could bind to that target sequence. Okay, and Cas9 would come and cut both strands of the DNA. This is called a double-stranded break. So not only one strand, but both strands. This is introduced at a specific place because the guide RNA, again, is complementary to that specific target DNA. It's binding in a complementary manner. Anyways, what happens is that the cell will try to repair the double-stranded break that you have caused here, but the repair is often error-prone. And therefore, a few extra bases are included during the reparation process, and a gene is then disrupted and deactivated because of that few extra bases. So yeah, this is how you switch off a gene. And this is kind of useful. Think about gene therapy. In our previous videos, we have talked about how gene therapy can only be applicable to recessive alleles, diseases that are caused by recessive alleles. And it has to be a single gene. And if it's a dominant allele that causes a disease, it's very difficult. But if we have figured out a way to use CRISPR-Cas9 in human beings in a safe manner, this could help us delete that dominant allele. Okay, we can introduce Cas9, a guide RNA that is complementary to that dominant allele, and then Cas9 can go and just cut that dominant allele, and therefore when the cell tries to repair it, a few extra bases are included, and therefore the dominant allele is switched off and you have cured the disease. How cool would that actually be? Now, other than that, actually, this technology is like very versatile, so you can even combine it with other enzymes to introduce specific mutations. So not like random bases can be added, but like specific bases can be added into there. It could be used in order to increase transcription with combinations of other enzymes, and even insert a gene that you want at a specific location, which is something like recombinant DNA technology cannot do. Gene therapy also cannot. Yeah, it will always insert genes at a random location. But using this new technology, we can insert gene at specific locations. It can even combine with GFP and act like a gene probe in situ. So basically, you can make it light up. So instead of using a gene probe in electrophoresis or gene probes in microRNA with microarray, which is kind of like outside the cells, you can just take the gene, take the guide RNA and Cas9, mark it with GFP, inject it into an organism, and the organism will light up wherever the gene is at. How cool would that be? That is in the organism, in situ. So yeah, this leads to many cool experiments and research, and it's kind of affordable. It's very versatile and improving, as we said just now, and therefore many things have come out of this. This is not legal to use in humans just yet. Okay, it's not legal to use in humans, but it's very legal to use it in animal research as well as plant research. And this is one of the examples. In this research, they are trying to end malaria with a gene drive. I won't explain it to you, but basically they are producing sterile mosquitoes by using this technology. You can check it out in the video linked here. Again, not in the syllabus, this is for information's sake. Now, of course, the biggest potential with CRISPR-Cas9, like I mentioned just now, is to cure diseases. So like genetic diseases caused by dominant alleles can be cured, even recessive alleles as well. Okay, you can even correct the mutation potentially. Maybe we can fix cancer that way. It could maybe cure viral diseases, because viruses do inject their DNA into our cells and modify our DNA, right? So using CRISPR-Cas9, maybe we can find a way to delete the virus from our DNA. That would be so cool, right? And potentially we can just inject the CRISPR in our body, and ta-da! It does the work for you. It just corrects all the DNA for you. Well, of course, this is not 100% effective just yet. Okay, CRISPR-Cas9 doesn't have a 100% success rate, okay, but it's much higher compared to other technologies where there's only 1% success rate. So yeah, it's exciting, and there's a lot of research now on it, and maybe we can even extend life expectancy and live longer if we do have this technology in order to cure all of diseases. But there are potential issues and problems, you know. What if CRISPR-Cas9, okay, is legal in human beings? What if in the future genetic editing, genome editing, is so easy. You can do it on any person and very quickly for very cheap. What would happen? So here are some ethical issues that are food for thought. You can watch the YouTube video here that talks about it in more detail. But here are two that I just want to highlight. A bit far-fetched, but good to think about it before it happens. If we can genetic edit with very little consequence, with very cheap, maybe there will be increased pre-selection of embryos forming designer babies. So maybe you can then use CRISPR-Cas9 to edit the genome so that your child has blue eyes, blonde hair, increased IQ, for example, increased muscle mass. Okay, and this is the same for super soldiers and biological weapons. So you can maybe genetically edit your soldiers to have more strength, more muscle building capacity, to be taller, to be bigger. Maybe even biological weapons. So maybe design a virus that could, cause a pandemic, but only you have the cure. Conspiracy theories. Okay, anyways, those are fictional at the moment, but it could be a problem if CRISPR-Cas9 becomes widely spread and very cheap and is not regulated by authorities. This becomes an issue. So yeah, but to be honest, like every single genetic technology we just talked about all have ethical and social implications, and I don't talk about them for fun. They are important as a human being, and also it comes out in exam. So yeah, don't disregard it. They're very important. So yeah, this is the end of the video, honestly, and this is the end of our A2 syllabus. Yay! I'll link all the relevant YouTube videos below. But just before we close, I just want to list the number of genetic technologies we've learned so far. So we have learned 5 genetic technologies in the syllabus. Number 1 is PCR. Number 2 is recombinant DNA technology. Number 3 is gel electrophoresis. Number 4 is microarrays. Number 5 is bioinformatics. All the applications highlighted in red are the important applications, so that might be what you'd be interested in. You realize that microarrays and gel electrophoresis has the P5 tag on them, which tells you that yes, they can come out in paper 5. Last but not least, I did teach you the CRISPR-Cas9 technology, but that is not in syllabus. That's just for fun, and it's incredibly exciting. I'm excited. Anyways, I'll see you in class. All the best for you two guys. Spam pass here. Bye bye.